Everyone. Thank you. My name is Andrea Sway, and I'll be introducing our speakers today. So we have our two speakers that are presenting today. First, we're going to hear from Mackenna Berg. Mackenna is currently pursuing a 4+1 master's degree in biology at Arizona State University with Sylvie Hupin. Her research interests involve understanding different mechanisms of resistance and has been working on a project with Culex tarsalis mosquitoes in Arizona, studying insecticide resistance involving various metabolic enzyme traits and genetic mutations. So go ahead and let her take the screen.
So yes, thank you. All right. Can everyone see everything?
Yep, looks great.
Awesome. All right. So yes, thank you for that introduction. Again, my name is Mackenna Berg. I'm with Arizona State University. Also working with Dr. Sylvie Hupin, and today I'm going to talk to you a little bit about what my master's thesis is on, and it's on the vector Culex tarsalis, understanding resistance mechanisms in Maricopa County and Arizona as a whole. So a bit about the Culex tarsalis mosquito, for those who are unfamiliar, they are the major vector for West Nile virus in the United States, and they are one of the most abundant species here in Arizona. They thrive in rural zones, agricultural areas, but also residential areas. So we've been seeing them a lot in different suburban areas, as well as farmlands and different riparian wildlife preserve areas. So because they are found pretty much all over, they are under insecticide pressure from agricultural spraying, public health and vector control fogging, as well as commercially hired insecticide use as well. So these are just a few pictures of where we find Culex tarsalis in Maricopa County. So that encompasses Phoenix, and we have a lot of suburban and city areas, but we also have some areas like the Tres Rios Wetland Preserve, as well as the Salt River right here in Tempe, Arizona. And then we also have a lot of places with artificial lakes as well, like this neighborhood called the Islands in Gilbert, Arizona. So you might be thinking of just Phoenix when you hear about Arizona, but we're actually quite diverse in landscape and ecology. So these are a couple of photos I took from some field work I did in Mojave County a couple months back. As you can see here, there's a lot more wetland reserve areas as well. And then this encompasses Lake Havasu. This is on our northwest side of Arizona. And then I also did some field work in Yuma this past month, a couple images as well. Yuma is home to lots of farmlands, as well as some urban areas and wetland areas as well. So I'm sure, as most of us know, that insecticide resistance is a huge threat to our vector control efforts. Each county is a little bit different in Arizona, but specifically Maricopa County uses truck-mounted fogging to keep our mosquito populations in control. Again, those who are unfamiliar, Maricopa County had a large outbreak of West Nile virus in 2021, resulting in 101 deaths. So we do a lot of surveillance as well, and then fogging is applied when we hit those thresholds. I'm sure, as everyone knows as well, that the repeated use of insecticides, specifically pyrethroids, has been found to confer resistance in a multitude of mosquito populations and species. The two most studied mechanisms of resistance are target site modifications and overproduction of detoxifying enzymes. So the goal of my project is to elucidate the insecticide resistant status of Culex tarsalis, both at the phenotypic, genotypic, and enzymatic level. And then along with this, to determine if there's a correlation between the lethal dose at 50% and the lethal time at 50%. So I want to just briefly discuss the methods that I'll be using. Initially, I'll have to collect the mosquitoes to understand what that looks like here in Arizona. So this is a map I have of current collection sites. I have one down here in Tucson, if you can see my mouse, that is hopefully soon on the way. So I'll be working with Pima County as well. But yeah, these are the collection sites I have so far. And I am looking to collect mosquito eggs or larvae so I can account for their age when I'm doing the phenotypic tests. So in places that I don't have any access to water, I will place these, they're cement mixing bins, and I just fill them up with water and then put hay pellets in them to incentivize the mosquitoes to lay their eggs. But in places that I do have access to water and can do it properly, I will collect any larvae or egg rafts that I can find. So the two pictures on the left are actually from the Salt River in Tempe, Arizona. And then the picture on the right is a photo from Mojave County. So I just wanted to briefly discuss the methods for phenotypic resistance testing that most people use. The three most common are the CDC bottle bioassay, the WHO tube test, and the topical application bioassay. Specifically, the CDC bottle bioassay is most commonly used as it's relatively easy. You can coat your bottles and bring them with you in the fields. In real time, you're able to collect mosquitoes via aspiration and do your assay right there at your field site. So it's very convenient. However, some studies have seen not the most reliable results with them and not as repeatable results. So I'd like to look at both the bottle bioassay and the topical application for my project. So those who are unfamiliar with the topical application bioassay, you take your group of mosquitoes from your field site and knock them down with CO2, and then you keep them on ice. We will separate the mosquitoes by species and then by males and females. So we're only doing this test on females. We'll separate them into groups of 20 to 25 and then put them in individual cups. We'll have 5 different doses plus a control group. So each cup will get a different dose. Then we will be able to weigh the mosquitoes per cup and then dose them individually on the mosquito's abdomen. After dosing is complete, they'll be placed back in the cup with some sucrose, and then we will assess for mortality 24 hours later. So these mosquitoes, we'd like to be age 2 to 6 days old, and then we'd like to repeat these bioassays at least 2, but hopefully 3 times per site. And then using this data with the different doses, we're able to determine the lethal dose at 50% mortality. And then I just want to briefly discuss the bottle bioassay for those who are unfamiliar. You have 1 control bottle and 4 bottles coated with insecticide. You coat those bottles, make sure it's evenly coated with acetone or your insecticide combined with acetone. And then you allow the bottles to dry for at least 3 hours, up to 24 hours. We will then introduce 20 to 25 mosquitoes per bottle and assess for mortality every 15 minutes over a 2-hour span. These mosquitoes will also be 2 to 6 days old, and they'll be tested from each site at the diagnostic dose for, again, for 3 replicates per site. So using this time points, we're able to determine the lethal time to 50% mortality. Then I just briefly wanted to discuss how we are determining the diagnostic dose and time for those who are unfamiliar. The CDC recommends a diagnostic dose and time associated with the vector you are testing for. However, for deltamethrin, the CDC does not have a diagnostic time available for the Culex tarsalis. So this you kind of need to create on your own. So I'm using an expected susceptible lab strain to see at what time do 100% of the mosquitoes die from the diagnostic dose. And with this assay, we found that at 90 minutes, 100% of the susceptible mosquitoes died. So that time point we will compare to all of the other collections. So let me kind of orient you because this is kind of a lot of information. So we have time on the x-axis and mortality on the y. Still have our susceptible lab strain in the dark green, and then the dotted dashed line for the diagnostic time. And then in the purplish blues, we have our 2 Maricopa County sites. So the first site, the darker blue here, is going to be our river bottom site. So those are from the bottle bioassay. We have seen that those have been the most susceptible out of all the collections. And then we have our Yuma County sites in the pink. So we see the site 2 is also closer to susceptible, kind of right around the same profile as the river bottom mosquitoes, but then we have a little bit more resistance in the other collection. And then we see the other site from Maricopa County that's in a more residential and urban area with a bit higher of resistance here, and then finally Mojave County with the highest resistance in the bottle bioassay. So now using that data, we want to determine their lethal time to 50% mortality. So we can estimate this by doing a probit transformation for the mortality and making a generalized linear regression model. And this, we have determined that the susceptible Culex tarsalis have a lethal time of 49 minutes. Here's a table with all of the other field sites collected. So again, as you can see, we have the RT482 with the second highest lethal time to 50% mortality, and the Mojave one site with the highest. Interestingly, again, the river bottom mosquitoes have close to the same lethal time, as well as the Yuma 2 mosquitoes. So once we have our bottle bioassay data, we also want to see what that looks like doing a topical application bioassay. Are we seeing the same trends? Is there something else that might be different here? So overall, we can kind of see the same trends happening in the topical application. So in the dark blue on the most left is our colony mosquitoes with the most susceptibility. And then it's kind of the same story here with the river bottom mosquitoes being closer to susceptible. And then with our Yuma mosquitoes, we actually have more similar patterns between the two than we did in the bottle bioassay. And then still with the most resistance with the RT482. I'm looking to do some more replicates of this as well to kind of see if we can get a better story or if it's going to kind of end up like this for our dose response curves. And then as well with this, we want to determine the LD50, so the lethal dose at 50%. Susceptible Culex tarsalis have a lethal dose of 0.038 nanograms per milligram per mosquito. So this is weight corrected for this dose. And then this is our lethal doses per trap. So the Mojave data I have right now is currently in progress. I'm finishing up data analysis on those. And then we also have our resistance ratio. So that's calculated by the lethal dose at the field collected site divided by the lethal dose for the susceptible colony. So this gives you a fold change of about how much insecticide extra you would need to kill these mosquitoes. So for instance, the RT242 is our river bottom site, and that's about a 5-fold increase of insecticide. So that's still not super resistant, but then we see some like the Yuma 1 collection with a 37. And then once I do these bioassays, I will separate the mosquitoes from dead and alive after the assay. So the bottle bioassay after the 2-hour mark and the topical after the 24 hours. Then I want to see if they have any knockdown resistance mutations. So this is a mutation in the voltage-gated sodium channel of the mosquito neuron. That's a 1 amino acid substitution that can reduce pyrethroid binding affinity and then prevent the insecticide-induced paralysis of the mosquito. So Culex tarsalis have been known to have a mutation at the 1014 locus on the voltage-gated sodium channel, and it's a leucine to phenylalanine substitution. And then I also want to determine if there's any metabolic enzyme activity that's being heightened currently compared to our susceptible control. So we will be taking the controls for both the bottle bioassay and the topical bioassay, determine on a population level what that baseline metabolic enzyme levels will look like. And then also maybe comparing mosquitoes that lived from both of these assays and seeing what kind of enzyme levels they might be producing compared to ones that haven't, compared to the susceptible controls. So we'll be using the colorimetric enzyme assays to determine this. Looking at 3 groups of enzymes, the cytochrome P450s, the glutathione S transferases, and then the carboxyl esterases, both alpha and beta. These 3 groups have been known to confer resistance to pyrethroids, but also other insecticides as well. And then hopefully using this data, we can do a correlation analysis on mean enzyme ratio and resistance ratio per site and kind of correlate these 2 mechanisms of resistance with their phenotypic resistance. And then finally, to answer that secondary question of the study, we want to see if the lethal time at 50% mortality and lethal dose at 50% mortality are correlated. So again, as I said before, many people use the CDC bottle bioassay, but there's also some people who are using the topical bioassay. And we kind of want to see, can we correlate these things? If some is resistant in the bottle bioassay, is that going to translate into what it's going to look like in the topical application bioassay? So this is a graph of the data that I have so far. It's pretty minimal, only 5 plots or 5 points on the plot, but I have the lethal dose, and this is the weight adjusted lethal dose on the x-axis, and then lethal time on the y. As you can see, R-squared value of 0.63, so not the best, and it's not significant, but it does look like there is a trend so far. Of course, adding more points, so hopefully working with Pima County and then adding those Mojave County points on here as well, maybe help us tell the story. And then as well as maybe working beyond outside of Arizona, seeing if this is something we see in other states across the United States. And then looking at not only the Culex tarsalis vector, but other mosquitoes as well. And then just some conclusions here. So my project in its completion will be assessing analysis for resistance at the phenotypic, genotypic, and enzymatic levels. I so far have determined some phenotypic resistance throughout Arizona at pretty much every site. Again, there's a potential correlation between the lethal times and lethal doses, but definitely more data is needed to draw any real conclusions. And then I think this work will also be super interesting to compare resistance mechanisms between Culex species. So at a multitude of these sites, there are other Culex species that are being found, not just Culex tarsalis. Like in Maricopa County, we have a lot of Culex quincefasciatus that we have data on their resistance mechanisms. So I think this would be super interesting to compare to see if they're kind of all conferring resistance in the same way, or if Culex tarsalis have a different mechanism of resistance than quincefasciatus and so on. And then also understanding how this technical resistance, so from the bottle bioassay and the topical bioassay, it can tell us, okay, there is resistant present and what's the lethal dose, but we're not actually sure how this works in practical resistance. So when our county trucks are going out to fog, how is this resistance actually impacting their spraying? So doing some sort of semi-field trial to relate the two, I think would be very useful as well. And that is all I have. Just wanted to thank the Hyben Lab, especially Donald Ward and Dr. Sylvie Hyben. Huge thank you to James Will at Maricopa County Vector Control for the help on this project, as well as Mojave County and Yuma County Public Health Department and their abatement district. And then just a huge thank you to PacVec for sponsoring this work and allowing me to collect some full data. And thank you. Thanks for listening.
Great job, Mackenna. Thank you. I think we have some time for questions. If anyone has questions, go ahead and raise your hand, or you can put it in the chat. I had a question actually. So can you reflect a little bit on the differences between your sites? I noticed the Mojave site had particularly high resistance. So is there any difference in treatment, or what do you think is causing some of that variability?
Totally. This is something I definitely want to look into. I don't have records currently of commercially what's going on. Their public health department does not fog on their own. It's all outsourced. So I think I would definitely be interested in looking at that. It's located right next to Lake Havasu. But not necessarily in an urban area. So I definitely think getting records to compare what's actually going on in the field will be helpful in the future.
Looks like there's a question from Scott. Go ahead.
Hi, great research. Hope it goes well. The pictures that you showed, I'm from Long Island, New York, Suffolk County Health Department. The pictures you showed, large expanses of water. Do they contain fish? We don't have tarsalis here, but we have, Culex pipiens is our vector for West Nile here. So, and they don't tend to do very well breeding in areas where there's fish. So, a lot of those large areas that you showed pictures of, I was just wondering, do they actually breed, or are there little offshoots that breed and where the fish can't get to them? So thank you.
Thank you. Yeah, most of them are found from offshoots, so I don't think there's much happening, breeding happening in those large pockets of water because there are fish present in mostly all of them. But more offshoots where the fish can't get, we've found most of the breeding happening. So
Yeah, so here, we try to open up those areas to allow the fish to access them. Is there any kind of mosquito management along those strategies?
I am not sure. I think that would definitely be a really good idea. Yeah, I'm not sure exactly what they do for those. I would imagine some sort of breaking up, but there's a lot of little pockets and just kind of depending on, a lot of these are located by rivers. So I'm not sure exactly how that impacts those pockets and how they fill up, but I definitely think opening everything up would help resolve that issue as well.
Thank you.
Yeah, thank you.
Great. Okay, well, if there are no more questions, we can move on. Thank you so much.
Thank you.
So next we're going to be hearing from Michael Riehle from University of Arizona. Hello. So Michael Riehl's lab is interested in developing new strategies for controlling mosquitoes and the diseases that they transmit. Specifically, they are interested in attempting to understand the basic molecular mechanisms regulating reproduction, innate immunity, metabolism, and lifespan in the mosquito. They're also interested in understanding the ecology of the mosquito Aedes aegypti, primary vector of dengue, Zika, and chikungunya viruses that represent a possible risk to Arizona. So he will be talking about age grading techniques for the mosquito Aedes aegypti. So Michael, you can try to share your screen.
Thanks, Andrea. All right. Can everybody see that? Okay.
That's good.
Thanks. Yeah, so I'm Mike Riehle. I'm down at the University of Arizona, so just down the road from ASU and Mackenna's work. So we're down here in Tucson, and our lab works on a number of different projects. Like Andrea was talking about, a lot of our work is based on genetically engineering mosquitoes and doing malaria work, but I've gotten involved in age grading of mosquitoes in collaboration with Kathleen Walker and Casey Ernst and some other people down here. And so that's what I'm going to be talking about today. So Aedes aegypti used to be present in Arizona up until about 1950, and then for unknown reasons, it left the area. And it was reintroduced in the mid-1990s and has since spread widely throughout the southern part of the state, including Maricopa and the Pima County where Tucson is. And we make wonderful habitat for it in the deserts in our backyard by putting out containers that can retain water and stuff like that. And then more concerning, the mosquito was reported in California back in 2013. And as you can see on this map here, it has since spread throughout a large portion of the state. And so it's really important to understand what these mosquitoes are doing, how long they're living, and how important of a disease risk they are for things such as dengue and Zika and chikungunya. So why do we care about mosquito age? So old mosquitoes, old female mosquitoes are the ones that are transmitting the pathogens, be it the virus or malaria parasites. Most of these have extrinsic incubation periods are in the range of 10 to 14 days or even longer. So this was a model put together by Scott O'Neill's group a while ago that estimated that there was, if you started with 1,000 mosquitoes and went with a mortality rate of, I think, about 90% per day, and you had an infection rate of about 6%, you can see the blue area over here are the mosquitoes that are capable of actually transmitting a virus. And so the way this works is basically the first 24 hours or so, the females are just trying to find a mate. Then day 2 and 3, they're trying to acquire that first blood meal. And if that blood meal happens to be infectious with a virus or a malaria parasite, Then the extrinsic incubation period is initiated. And this incubation period, again, is dependent on what pathogen you're looking at, as well as temperature and other factors, but typically it's fairly long, about 10 to 14 days. So we're assuming a 10-day incubation period here with this generic virus. And so the first mosquitoes that have virus in the salivary glands and could potentially transmit it back to a person are almost 2 weeks old. And this is pretty old for a mosquito, especially in the field, where not only are they just having general senescence, but they're also facing all the hazards and predation of being out in the field. So it's these mosquitoes here that we're really concerned about, these older geriatric mosquitoes that have had the potential to have the virus in their salivary glands. And this is, of course, a best case scenario because there's no guarantee that first blood meal is going to be an infectious blood meal. The female might not pick the virus up until the second blood meal or the third blood meal. So really understanding how many of these 2-week-old and older mosquitoes are out there can go a long ways to informing us on what the disease risk and the transmission risk is. Okay, so how does knowing mosquitoes' age help us? Like I said, understanding the percentage of the mosquito populations that actually represent a risk to us, tracking that population over time throughout a monsoon season here in Arizona or whatever the typical mosquito season is in the given area. You can use this information from field collected data to inform models and estimate risk. But in order to accomplish this, this is what we really need. In a perfect world, we'd have a simple, accurate, cost-effective, and high throughput approach to accurately age grade mosquito. And big hint here, we don't have that yet. So, I mean, we have some of these. We have some that are fairly simple. We have some that are reasonably accurate, but we don't have anything that's really practical in a applied sense. Okay, so there's a number of different techniques that can be used to age mosquitoes that have been described over the years. You have stuff like cuticular hydrocarbons, ovary tracheation, stuff like that. I'm only going to talk about 3 of them today, and the first 2 are what we started with in the lab. And the first one is ovary tracheation. So what this is a very small portion of a mosquito's ovary. And these darker areas on these pictures are the trachea. And what happens is when a mosquito emerges from the pupal stage, she has trachea that have these little coils on them that you can see here. And the reason for that is these trachea don't expand. So they have to be able, when the ovaries swell up with a full clutch of eggs, these trachea need to be able to unfurl so they don't break when the ovary enlarges. Problem is when the eggs are oviposited, the ovaries contract back in size, but they don't form these nice little tracheal skeins. They just form this spaghetti mass of trachea that you see in this Paris mosquito that's gone through a reproductive cycle. And so if you carefully dissect out the ovaries from mosquitoes, if you see these tracheal skeins, you know that they have not gone through a reproductive cycle yet. They haven't produced a clutch of eggs, they haven't taken a blood meal, and they have absolutely no chance of transmitting a pathogen. These have taken at least 1 blood meal, so they might have picked up a pathogen, we don't know. Okay, so what are the pros and cons to this approach? Well, if you have somebody that knows what they're doing and you can very carefully, you can't stretch or pull these ovaries at all, or you'll pull those tracheal skeins. But if you have somebody that's skilled at this, the accuracy can be in the 95 to nearly 100% as to whether they've gone through a reproductive cycle or not. But to get to this level is technically challenging. It takes a lot of skill and time to learn how to do this. The other big problem is this only indicates whether mosquitoes have completed a reproductive cycle or not. And so based on some studies we did previously, at least in Arizona, the mosquitoes are quite successful at getting that first blood meal in their first few days. So typically, if we still see these tracheal skeins, we can know that they're very young mosquitoes. But it doesn't tell us much beyond that. So that's approach 1. Approach 2 is the expression patterns of age-associated genes. And there's been a few studies out there. There was one by Scott O'Neill's lab that looked at 10 different genes that either increased or decreased in a predictable manner as the mosquito aged. And we tested all of those. The one that worked for us is this one, SCP1, sarcoplasm calcium binding protein 1, and it decreases in expression in a predictable manner as the mosquito ages. The problems with this one is the accuracy is fair, somewhere between 85 and maybe 90%, as far as grading mosquitoes into 3 classes. So we broke them down into non-vectors, which we considered less than 5 days old, potential vectors, which were 5 to 14 days old, and then high-risk mosquitoes that were 14 days plus. The other problem is it's expensive. By the time you isolate the RNA, do the cDNA synthesis, amplify this up with real-time PCR, and including the person's time, you're probably looking at about $8 to $10 per mosquito. So it's good, but it's expensive. Okay, so enter near-infrared spectrophotometry, or NIRS. So the way this works is you have a mosquito and you hit it with a beam of light, and you measure the near-infrared wavelengths, which runs roughly from 500 to 2,500 nanometers. And depending on the bonding in the mosquito, it's mostly looking at oxygen, hydrogen, nitrogen, hydrogen, and carbon, hydrogen bonds. You have different levels of absorption and reflection at each of these individual data points. So the near-infrared machine collects all 2,000 of these spectra in just a few seconds and looks at how much is reflected back into the machine. And so you end up with a curve like this. The problem with this is you can't point to any particular point on these graphs and say this is water or this is some specific molecule. You know, a change in water composition results in changes throughout the curve. So you really have to use machine learning to try to identify patterns that are associated with different aged mosquitoes. So the downside to this is there is a fairly large initial upfront cost for the machine. And the accuracy, although we don't know truly what its accuracy is yet, it's almost certainly lower than the other techniques that we've looked at. But once you've actually purchased the machine, the cost is negligible. I mean, you're hitting these with a beam of light and, you know, there's no other resources needed. You might have to replace a halogen bulb every year or two. So it's virtually just the person's time. And it's very simple to do. I mean, anybody can put a mosquito under this light, hit the button, and it'll spit out a predicted age. Again, how accurate that age is up in the air. Okay, so this is what we're looking at to see if it'll work. And to test this, we developed 4 different models from 4 mosquito populations. So these first 2, the frozen model up here and the semi-field model, are both based on well-established laboratory colonies. So it's the University of Georgia laboratory strain, or the UGAL strain. It's been in culture 50 to 60 years. You could make an argument of whether or not it's even still a mosquito after that long in culture. But we developed these first 2 models over this. And the frozen just means we immediately froze the mosquitoes. And then for the semi-field, we actually, once they emerged as 1-day-old adults, we actually reared them out in my technician's backyard for 18 days and collected various ages. But we were concerned that these weren't going to be reflective of mosquitoes in the field. So we collected mosquitoes from Tucson, established a colony, and at the F2 stage, we collected the mosquitoes for age grading. And then we did the same thing up in Maricopa County, which we collected these mosquitoes from a field site that we did. We had a larger project that was being done at, and so these are directly reflective of the mosquitoes I'm going to be talking about in a little bit. All right, so what happened? So we took anywhere from about 500 to 700 female mosquitoes from all these different time points, and 80% of them, so 4 out of every 5, we used to generate the model using machine learning software. And then we held 1/5 of them back, which is the test set here. And those were the ones that we then validated the model with. So they weren't actually used to generate the model. And so that's what the calibration set was used this number used to actually make the model. And then the test set is the black triangles that were used to test the models. And overall, if you look at this, this is pretty good. It's pretty successful. And for all the models, it did a pretty good job of age-grade mosquitoes, with one huge caveat. These are all lab-reared mosquitoes. I mean, even these were reared in our insectary under ideal conditions with ample food, no stress. They weren't crowded, consistent temperatures and consistent humidities. So in the lab, this works great. So the question is, does this actually translate to the field? Well, that's a tough question to ask, actually, because we don't have a technique for age-grading mosquitoes at any level that is 100% accurate, regardless of cost or time or whatnot. So all of the tools that we have are not necessarily accurate. So it's difficult to say exactly how old that mosquito is in the field. So I was mentioning this larger project that was done up in Maricopa. It was actually done in Gilbert and Chandler, Arizona from 2017 to 2019. And a little over 2,000 mosquitoes were collected, and we collected NIRS spectra from all 2,200 females. And again, these were wild-caught females, so we have no idea how old they are. So this is the study site. So again, Phoenix right here. This is a suburb down in the southeastern part of the Phoenix metropolitan area. And this was the experimental setup. It was divided into 1 mile squares. And then we had matching sets where we could do a treatment and control. Again, this is for another project, so not really relevant here. Okay, so all we could do was use our other age grading tools to try to get an assessment on whether the predictions from the NRS age model seemed more or less reasonable. So the first thing we started with ovoretracheation. And with ovoretracheation, again, all we know is did they go through reproductive cycle or not? We don't know how old they actually are. But in general, all the nulliparous mosquitoes, the ones that had not gone through a reproductive cycle and still had those tracheal skeins, were always significantly younger than the Paris mosquitoes. Not a huge surprise, but at least for the NIRS age predictions, you know, that suggested that it was identifying older mosquitoes in those that were Paris. Not a terribly strong argument, but that's what we did. And out of all of these models, the frozen model tended to overpredict the ages a little bit. Semi-field in Tucson underpredicted the age, and the Maricopa mosquitoes seem to be most accurate. And again, this Maricopa model is being tested against mosquitoes collected in Maricopa County. So that's a good indication that you're going to need to develop models based on locally collected mosquitoes. Okay, so then we tried comparing it versus the SCP-1 analysis. And in this case, again, we have our 4 models, frozen, semi-field, Tucson, and Maricopa. 2 different years of mosquito collections. And yeah, there's some cases a trend up, but there's a lot of messiness down here, especially at the younger ages. So the problem here is that you have 2 techniques, both of which have a large degree of inaccuracy. So the SCP-1 analysis, 85 to 90% accurate, plus or minus 3 days of age. The NIRS is probably closer to plus or minus 5 days. So you're trying to validate 2 imperfect models with each other. And again, there's a suggestion that it's working, but you're never going to be able to look at any individual mosquito and say that mosquito is X days old. Okay, so what did we learn from these initial studies? So again, like I said, NIRS models are unlikely, I would say, are never going to be able to predict the age of individual mosquitoes unless there's some dramatic change in technology. But I would argue they are useful for looking at the population age structure over time. So if you're trying to see, as the season goes on, are you having more older mosquitoes coming out, or are you having an influx of young mosquitoes that have recently emerged? This will give you an idea of what's going on over time. And because you can collect large numbers of mosquitoes, I mean, scanning 1,000 mosquitoes is absolutely doable and inexpensive to do. So you can get large numbers to look at these that help give you a better idea of what's happening at a population level. And this will, of course, allow us to predict peaks of older high-risk mosquitoes. Very simple, cost-effective technique. But we're really nice to have some sort of age grading tool that's near 100% accurate, regardless of the cost, just to validate both the SCP-1 and the NIRS techniques. So, okay, so that's talking about age grading mosquitoes. But we're trying to use NIRS as something else too, which is not necessarily determine their age, but to determine the number of blood meals that they've consumed. Because at the end of the day, this is arguably more important than how old they are. If you have a really incompetent mosquito that can't get that first blood meal for the first 2 weeks, it's not going to be a vector, even or an important disease vector, even though it's an older mosquito. But if we know that a mosquito has blood fed 3 times or 4 times or 5 times, then we can get a better measure of how likely they are to have picked up a pathogen and what risks they might present for disease transmission. Okay, so this looking at blood meal is measuring physiological age rather than just straight chronological age. So, how far along are they physiologically? The problem with this is there's really limited options for measuring the number of reproductive cycles from field collecting mosquitoes. There's basically one, and this is this Polividova technique, which is ovarial dilation. So what happens here is if you look at the individual follicles in the ovaries, you can identify these little dilations if you inject oil into the ovarials and allow these dilations to swell up a little bit. That helps you see them. The problem is, this is incredibly technically challenging, time consuming, and is not going to be practical at any level for applied use. But it is a tool that could be used for validating an approach. So our question was, could we use near-infrared spectrophotometry to predict the number of blood meals that a mosquito has gone through? Okay, so our very preliminary experiment looked at the number of blood meals. So we had mosquitoes that were given no blood meals, one that was given a single blood meal at day 4, one group of mosquitoes that were given blood meals at day 4 and day 8, and then another group of mosquitoes that were given blood meals at days 4, 8, and 12. And then they were all harvested at the exact same age. They were 16-day-old mosquitoes when we harvested them. So we removed their age from the equation in this case. And what we saw was pretty exciting. We saw a very nice upward trend in the number of blood meals that were predicted by the model. So this is a number of known reproductive cycles. They either non-blood fed, 1 blood meal, 2 blood meals, or 3. And this is what the NIRS prediction model for the number of blood meals came out with. Now, there's still variability. I'm sorry, I forgot to remove the labels for it. So it's a bit of a mess, but these are all just individual mosquitoes. But you can see maybe 0, 1 can't be discriminated very well, but certainly by the time you get to 2 or 3 blood meals, you're getting fairly large differences from the 0 and 1 blood meals. So this was encouraging. But we had this elephant in the room that we know age is impacting the NIRS spectra as well too. So the question was, okay, do we do our blood meal analyses? Are we just seeing differences due to age, or is that going to mess up our predictions? So we came up with this entire battery of different treatments that we were going to do. So these were mosquitoes that were fed either one time, so at 4 days, 8 days, 12 days, or we actually never got to the 4th, so we're just doing the first 3. So 4 days, 8 days, and 12 days, then 2 blood meals with all the combinations, 3 blood meals, and 4 blood meals. But again, we didn't reach 4 blood meals. We ran out of mosquitoes. So I tasked my graduate student and one of their undergraduate assistants with this, and after much work, what they found was they still saw pretty much the same thing, and it was largely independent of age. So this is similar to the initial graph. The blue indicate the mosquitoes that were used to generate the model and predicting 0, 1, 2, or 3 blood meals. And then the yellow was the mosquitoes withheld from the model to validate it. And then over here is just looking at the different ages of the 0, 1, 2, or 3 blood meal mosquitoes. And there really was not any big age difference that jumped out in these predictions. So that was exciting. It didn't seem like age was conflicting with things. And we actually went and used all those mosquitoes from that frozen model. We still had all those spectra saved. And the nice thing is once you have the spectra, if you update the model or if you make a new model, they're still there. You can still reuse them. So these are 760 non-blood fed mosquitoes of all different ages. And so what we wanted to see was that everything was reading around 0 and that there wasn't linear regression going upwards as the mosquitoes aged. And in fact, what we saw was for the most part, they were hovering all around the non-blood fed status. 14 days, 20 days, they were a little higher. But overall, it was pretty encouraging. Okay, so real quick, I know I'm running a little bit over, but wanted to mention real briefly what we were working on with Chris Barker's lab and his student Dalton, who collected recently colonized mosquitoes from 2 locations in LA, Lakewood, California, which is down here, which is coastal and cooler and more humid, and Tujunga up here, which is inland and hot and dry. They established these colonies recently because everything else that we had done so far was, again, with that Yuga colony that had been cultured for a long time. So we wanted to see if this blood meal stuff held up. Real quickly, we did an age grading analysis too, so that worked fine with both of these colonies. So I'm not going to dwell on this in the interest of time. But the exciting thing was the blood meal seemed to also be highly predicted. So yeah, in this case, it was either non-blood fed, 1 blood meal or 2 blood meals is what was available at the time. And again, arguably even better than the Egal strain, we were able to discriminate the number of blood meals for these types. So this is something that's ongoing, and we're definitely expanding on all of this work. So to sum everything up, NIRS age grading works great in the lab. Field is still a question mark. There's a wide range of variability, however, even in a lab mosquito, so it's never going to be capable of age grading individual mosquitoes, but it could be important at population age structures. Quantifying reproductive cycles with near-infrared spectrophotometry looks promising, and the nice thing is it can be accomplished in concert with the NIRS age grading. So you get these spectra, you can run through 1 model to predict the age and another model to predict the number of blood meals. And then thanks to everyone who helped with this, specifically in my lab. Teresa Joy did the initial NIRS age grading work and the SCP work. Zoe right here and Raya right here were involved with the age grading and blood meal analysis, but most of that was done with Adrian, which handles all of our mosquito rearing. And then in Chris's lab, Dalton here did all of the work on the local mosquitoes. And then obviously all the field collecting mosquitoes were done by our collaborators here. And if we have any time, I'm happy to take any questions.
So interesting. Yeah, we do have some time. If people need to hop off, they are free to, but looks like we have about 10 minutes for questions.
Ben?
Let's see. I have a question. Oh, Chris has a question. I'll let you go first.
I was just curious about what you would view as the main molecular changes to cuticle after blood meals. Because I've always naively thought of it as once the cuticles form sclerotized, that it would be pretty static. And it looks like from what you're seeing that maybe there are molecular changes happening.
Yeah, I mean, what exactly is happening? I mean, again, this is, looking at the excitation of these hydrogen bonds between different molecules. So it's a little difficult to say exactly what's going on, but my guess would be it's the cuticular hydrogen carbons and the composition of them on there that are changing over time. There's a wealth of information out there showing that those change in fairly predictable manners as mosquitoes age. It's just that the approaches to trying to characterize those are very difficult to do. So I'm going on the assumption that that's what's happening.
Thanks. Great presentation.
Yeah, and I'm sorry about the background noise here. There's like a large truck, but I think it's a fascinating method, and I'm wondering because you showed that there was an increase in or you could predict the age in the lab. using this method, and then also that it was sort of age agnostic in the blood meal analysis. And so I'm wondering, is it measuring different things, or is it just a training model that is able to tease apart those two different signals?
Yeah, absolutely. So it's definitely measuring different things. That was our big concern. Was we did these initial blood meal or reproductive cycle models, and we thought there was going to be a fairly strong chance that when we added age into it, a lot of the changes we were seeing was just due to the fact that there were older mosquitoes, but that didn't happen. So it's a bit of a black box. You know, you take all these spectra, you tell the software that these are the different ages, and you send it in for the machine learning to try to parse out which parts are important, which ones aren't. But they don't tell you at the end of the day what they are. But clearly we did not see an interaction between age and blood meal. So that should allow us to use those both independently on the same spectrum.
I see. So that initial image you showed of the spectrum, the full spectral reading, it might be using different aspects of that signal for different training models.
Yeah, absolutely. And actually, when you're developing the models, you test different sections as well as the whole spectra, because sometimes some of the regions of the spectra can introduce noise and whatnot, and so you're just you just keep trying to tweak the model again and again to see if you can improve on it slightly. And like I said, the spectra, as long as you don't lose the data on your hard drive, they're always there. So if you make a better model, or if you get more mosquitoes that you can use to generate the models, if you double your sample size or whatnot, you could easily make another model, then run those same spectra through again and get a new prediction. So
The question in the chat from Benjamin. Could putting down known age lab mosquitoes in larger outdoor cages help give some more field applicability to the training data?
yeah, absolutely. And that's what we tried doing a little bit with that semi-field thing, but we didn't actually use significantly larger cages. So they were at least exposed to the environment, but they were still in a fairly constricted area. So yeah, having much larger cages out in the field could get you better information. Absolutely.
And then I have another question. Do you think this would work on ticks as well? You look at age.
We're always willing to try. That's a nice thing with this. I mean, there's no real expense besides time to test these ideas out. So I think it's reasonable to predict they would. Yeah.
With some of your ticks, Andrea, do you measure that in years or days or weeks?
I mean, that's the thing, is when you see a questing tick, you don't know if it's from this season or last season. You assume it's this season, but there's always been a bit of a mystery because we can't tell.
Yeah. The biggest problem is you need large numbers. So if you only have 20 or 30 ticks, that's going to be tough to glean anything from. But if you have a few hundred, I mean, especially if you're just looking for last year versus this year, or whatnot, a few hundred ticks at each point, we could certainly try that.
I will be in touch then.
Excellent.
Let's see, any other questions? It's like, if not, we can let everyone go a little early. I did want to just remind everyone, unless my would know, we have another set of talks in 2 weeks on, on May 27th, we are hearing from Talia Wong from UC Davis, and also from Mahmoud, and I can't find his last name, but from CSU San Bernardino. So please join us for that as well. So if everyone can just join me for a round of thanks to our speakers today, Mackenna and Michael. Thank you so much.